Hello everyone, welcome to WPI and BME. Congratulations on being accepted to WPI. My name is Professor Shaktikumar Ambady. I have been with the department for 10 years. I'm a teaching associate professor and my background is in veterinary medicine, molecular genetics, molecular biology, and regenerative medicine. Since I'm a teaching professor, I don't do my own research, but I do teach 2 courses and then take care of the advising. I advise a lot of students, including pre-med and other pre-health advising. I also coordinate the MQP in the department. So I'm going to talk to you about my courses, how I teach, and what my students learn in my 2 courses. So I teach 2 courses in the department. One of them is BME 3111, Physiology and Engineering. It has a lecture and lab component. So during lectures, depending on the class sizes, I try to introduce group projects. In here, students are given a challenge and they will try to solve it. And then the groups will exchange notes, and then there will be take-home exams from these discussions. The other interesting and exciting component is the lab component, where students are excited to do live rat surgeries. We share this lab with the Department of Biochemistry. The students also learn LabVIEW to collect data in real time. The second course is Cellular Engineering, or BME 3813, where the students learn the basics of cell culture and imaging techniques. They learn the importance of culture media, the pH of the media, the culture conditions, and why it is important to follow sterile techniques to prevent contaminations of bacteria, yeast, fungus, or mycoplasma. They do short-term and long-term projects. So this is an example of a long-term project they do for the whole term. So they are studying the cell growth dynamics in 2 different culture conditions, one containing 10% fetal bovine serum and 5% fetal bovine serum. And these are from student presentations. Another exciting project is C2C12 cells, a mouse myoblastic cell line that can differentiate into twitching myofibers in 7 days. So on the top left, you have the undifferentiated cells, and when they are put through a protocol, you see on the bottom right, fused cells with the nuclei aligned in the center. And if you really induce them to twitch, they can actually twitch. Another project, short-term project, is immunocytochemistry, where they learn how to detect a protein inside a cell. In this case, actin cytoskeleton is detected using phalloidin Alexa Fluor 488 and detected by fluorescence imaging. Students also get to do 3D culture. So here is a fluorescent labeled cell line in a 3D culture on day 1, and then by day 9 these cells have grown, and as you can see, the cells go in and out of focus because they are dispersed in a 3D gel. This is a non-fluorescent cell, but then tagged with a nuclear stain and imaged using the fluorescent microscope. The students also learn to characterize the surface properties of cell culture substrates by looking at contact angles of liquids. And this will tell the students as to whether a surface is hydrophilic or hydrophobic, meaning whether the cells like to grow on the surface or not like to grow on the surface. And then they use this principle to coat a material called polydimethyl siloxane, or PDMS. And they coat it with polydopamine, collagen, polydopamine and collagen, plasma treated, or plasma treated plus collagen or polydopamine, or other materials that we have at hand. And so this is another independent project they do over a period of 2 to 3 weeks. Okay, so I hope that this gives you a rough idea as to what these 2 courses offer, and I'm more than happy to discuss these courses if you have questions. Again, congratulations, and I hope to see you very soon. Please don't hesitate to contact me anytime you have questions.